Hello, everyone, and welcome to today's keynote presentation titled Tracking Social Behavior and Its Neural Properties in a Smart Aviary. This webinar is part of the 12th annual event in the Neuroscience Virtual Event Series. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Now, before we begin, I would like to remind everyone that today's event is interactive. And we encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing the presentation, just click on that Help Desk button from the lobby within the event. And finally, as a reminder, today's presentation is educational and it does offer free continuing education credits. Simply click on the continuing education credits image located at the right of your screen and follow the process to obtain your credits. Okay, everyone, now let's get started. I would love to present today's speaker to you, Mark Schmidt, Professor of Biology and Neuroscience Program at the University of Pennsylvania. For a complete biography on our speaker, please find it on the left of your screen. Mark, welcome. You may now begin your presentation.
Thank you, Christy, for this very kind introduction. I'm very pleased to give this talk. What I wanted to do is walk you through a new direction that my lab has taken that I'm very excited about, and I'll show in the next slide. And I should mention there is no cursor, so I'm going to have to describe with my words what part of the slide I'm talking about. So what we did about 6-7 years ago, and this is an idea that I've had probably lingering in the back of my head for about 20 years, is we built a large aviary, which we call a smart aviary, because it's equipped with 10 computer vision cameras and an array of 24 microphones and other devices, where we aim to record the group dynamics of a flock of about 15 or so birds, males and females. And we're particularly interested, as you can see in the lower panel, to record this through the entire breeding season. And in the beginning of the breeding season, behaviors are really intense. Males are courting females. Females are paying very close attention to what the males are singing, how they're singing, and they're basically choosing their mates. And this is a really important behavior that's been, selected for through sexual selection over millions of years. And so males are working very hard to attract the females. And we think that behavior is really interesting and important, and ultimately leads to reproductive success, which is the ability of one generation to propagate its genes to the next generation. So the types of behaviors that are happening there are really important, and we're interested in the dynamics. Not only are we interested in the dynamics of the behavior, but we're also eventually interested in the neural mechanisms that underlie some of the choices that females are making, or the choices that males are making in the types of songs that they produce. And so, as you can imagine, this is a daunting task to try to record all this activity in freely moving birds in a naturalistic environment. And this then, leads to my introduction to my team, which is a transdisciplinary team. And this is the only way we can approach these types of questions. So here are my three very close colleagues. To the top left is Firuz Aflatooni. He's an electrical engineer, and he's working on developing wirelessly powered and transmitting neural devices. My friend Kostas, bottom left, is part of the engineering program here, and he's a world specialist in computer vision. And then Vijay Balasubramanian to the right is a professor of physics, but also computational neuroscience. And so he is really working with me very closely to try to analyze all the data that we'll be collecting. But before I go into the project, I think it's interesting to ask, what is the history of neuroscience, and why might it be interesting to study in a naturalistic context? And obviously, this does not just happen. Technology is going to drive a lot of this. So a lot of the tools we now have with artificial intelligence and all the different new technologies, improvements in computer vision and microphone arrays, now offer this ability, which we couldn't do, obviously, even 10 years ago. So what I want to do is walk you through the history of neurophysiological processing in the brain. And I think it's an interesting way to actually look at this. So neurophysiology in sort of complex vertebrate brains started around the 50s. And this was with the development of electrodes, very thin wires that are insulated except for the tip we can stick in the brain. And again, the brain doesn't have any sensory terminal, so it does not hurt. The animal does not feel the electrode going into the brain. And you can put the electrode and you can map out different responses for neurons, groups of neurons, or single neurons. And on the picture on the left, on my right, in this preparation, this was seminal work by David Hubel and Torsten Wiesel that they did in the early 60s, which really gave us an insight how the cortex is organized. And so they tracked responses in primary visual cortex to different stimuli projected on the screen. And it was really valuable information to understand how the brain is organized. The advantages of these preparations are that it's a very stable preparation. The animal doesn't move. And something that's maybe missed, but I think it's an important point, it's a single behavioral state. The animal is anesthetized, and it's not awake, because when you're awake, you could be attending, you could not be attending, you could be acting. And so that adds a lot of complications, which you don't have to worry about in an anesthetized animal. But what are the weaknesses? Well, there's no active sensing. As you know, we look at the visual world, we're constantly moving our eyes. If we touch, we're moving our hands to actually feel the surface. So that's important when you look at sensory processing, that you want to look in an active way. And obviously, you cannot study motor control under these conditions. So around the 70s, people started looking at ways to study motor control. And what they started realizing is that you could maybe work with awake animals if the head was fixed. And so this was first pioneered in monkeys, and then eventually now in rodents, as you can see on the bottom right. And this is a technique that is still used. Even the anesthetized technique is still used. It's great for mapping circuits in the brain. What you can do here is you have a stable brain, so you can still map the circuits out. But what you can also do is you can have the animal respond to a cue, for example, as you see in the monkey, and make a decision. And you can record activity of the muscles at the same time as you record activity in the brain. And so that allows you to study behavior. And stimuli can be controlled in exquisite ways. What this does not allow you to do is it does not allow the animals to interact with the environment, and they're not able to interact with other animals that might be present. And one other weakness that's going to be true for all techniques I'll talk to in the next 4 slides is that the behavioral state now is variable. The animals might be falling asleep, but they might also be awake. And if they're engaged in the task, their awake state might be different depending on whether they're getting rewarded or not rewarded, for example. And I should say that this approach is used in contemporary neuroscience all the time. As you can see with a mouse, we can put on a head-fixed mouse, we can bring a microscope and we can do two-photon imaging of cells that have been transfected with viruses that make them glow in the dark and respond to neural activity with changes in calcium, for example. But you can even study interesting behavior here, and just in the context of vocal communication. This is work by a friend of mine, Steve Eliadis, when he was in Xiaoxing Wang's lab that was published in Nature, where on the top right, you can see a call that these monkeys produce. So these are head-fixed monkeys, but they produce these phi calls. And we can see in black, it shows that during the phi call, there are fewer little black tick marks, which tells you that the auditory cortex is pretty much silent when it listens to the call of the animal. So when you're talking, for example, when your brain hears your own speech, it is pretty much silent. However, as you can see in the red, if you manipulate what you actually hear, if you introduce an error to the sound that you're producing, your brain is now responding very strongly. In other words, the auditory cortex acts as a very good error detector, which is going to be important eventually for correcting speech patterns. So this was in the 70s. And then in the 90s, we started, having small chips that we could put together. And this is work that I did, for example, in the top right, in songbirds, where I'm recording from individual zebra finches, even interacting with another zebra finch in an adjacent cage while these birds are singing. This has also now been done in rodents, and you can see this mouse is carrying a whole load of really interesting things. Sometimes you can even have put microscopes, miniature microscopes on the head, and you can image the brain. So this is great. You can now look at interactive behavior between two individuals, and these behaviors are more natural. But there are challenges here, as you'll see in the next slide as well. So the stimuli cannot be controlled because the animal is just doing what it does. The behaviors are variable, as I'll show you in the next slide. But the one disadvantage here is that in terms of social interactions, you're still limited to dyads. So dyads is two individuals talking to each other. And so the full range of behaviors is not really on display. And what this approach showed us in many different systems, and I'm just showing you an example in the songbird system, is remarkable. Here are two different conditions. You can see on the top of the figure on the right, there's a little male with the orange pouch on its cheek singing a song to a female. This is a female-directed song. And the male on the bottom, just below it, is just a male singing by himself. And you can look at the acoustic signature on the right of those diagrams, and the song looks essentially identical. And to us in the field, we thought they were identical. Until we recorded activity in the brain, you can see when the male sings to the female, is that the little tick marks, which represent action potentials in individual neurons across 100 trials, so the bird is singing 100 times the song, you can see there's a fair amount of structure from rendition to rendition. But if he's singing by himself, that structure is lost. So that was really interesting. So the bird sings the same song, but when he sings to a female, it's very structured. When he sings alone, it's not so structured. And then that made us actually go back to the song itself. And you can see on the little histograms to the right, you can see that one is more narrow than the other. And it turns out that when the male sings to himself, his song is a little bit more variable. It's the same song, but it's just a little bit more variable. And the song, when he sings to the female, is really the performance song. That's what he's trying to use to impress the female. So it's incredibly stereotyped and very tight. But this approach forced neuroscientists, and so neuroscientists, we're trained to kind of really look at the brain and really focus on what we want, is we realize that we need to learn more about behavior. And so I see a lot of neuroscientists, including myself, scrambling towards understanding behavior on a more behavioral ecology perspective. And 4 years ago, for example, I actually went to my chair and I told her, I said, I really would like to teach animal behavior because I really feel like I need to understand animal behavior much more. So one thing I want to introduce you is that, so this kind of led to the next stage of type of research, which is where my smart aviary comes in. And I'll call it neuroscience in freely behaving animals in a naturalistic social environment. And what do I mean by that? I mean, ideally, animals in the wild, and we can try to understand their behavior in a very quantitative way, and eventually record their neural activity to understand brain patterns in completely or as completely possibly non-invasive as possible. And so what is remarkable about this approach, though, is that there are behaviors that are never produced in the laboratory that you see in the wild. And I reached out to my friend Steve yesterday, who studies marmosets. And as you can see a picture of the 3 marmosets to the right, if you look at the number of calls that they produce, you'll see that if you take a restrained monkey, this is the head-fixed monkey I was talking about before in the example, is it produces occasional fee calls and sometimes alarm calls. If you take 2 monkeys, both head-fixed and allowed to call to each other, they will only produce fees. Now, if you put 2-3 animals in the room, now you increase the number of types of calls. And if you look at the home colony where there's freely moving and interacting, now you start having 5 or 6 or 7 different types of calls. But in the wild, you have many more calls. For example, a snake alarm call you're never going to see in a laboratory setting. So what that says is that studying animals in a naturalistic environment is really advantageous because you start seeing behaviors that are natural, that are maybe driven by the social context in which they find themselves that you never see in a laboratory situation. Obviously, there are going to be advantages, and those are the ones I just described to you, but there are also going to be disadvantages. But before I get to the disadvantages, what I want to say, you might ask why. Why do we want to study? I told you that it uncovers a number of behaviors. Why do you want to study certain animals in a naturalistic environment? And here I want to read a statement that I wrote in a recent grant that I submitted to the NIH. And we can think of this as a working hypothesis. So the way I'm thinking about it, and this is true for many different socially gregarious species. So in the top left, we have monkeys, then we have cowbirds. In the second panel, then we have bats, and then we have naked mole rats. Is that the nervous system of social species has evolved to perceive and evaluate signals within a social context. And therefore, social information must impact how the brain processes information. In other words, we're missing a lot by not looking at the brains of social animals, by looking at them only in a small box, or maybe interacting with one individual and not in a larger group. And so you might say, okay, maybe you're right, Mark, maybe you're not. But it turns out there are a few hints that this might be interesting. So I'm calling them some surprises. Again, this is a very new technique. So on the left panel here, we have two interacting mice where Heijie Wang from UCLA was recording and with his student Kingsbury, and he noticed, or they noticed, that the brains of two mice coming close to each other and interacting became synchronized. And synchronized in I won't go into the details, you can think of their brain waves are oscillating at the same levels, as shown by the orange wiggly lines and the green wiggly lines. And then a little bit more recently, by the laboratory of Michael Yartsev at Berkeley, who's really one of the pioneers of really pushing the envelope of looking at animals in a social context. He noticed, you can see the little diagrams in the bottom, like a little green head of a bat and a blue head and a gray head and orange head, is that if you take bats that are not really living together and you present a call to them, is that their brains are not very active. And if you bring them together physically, there's not much synchronization. But if you take bats that actually live together in small groups, their brains synchronize much more. So this suggests that there's something about affiliation between individuals in a group that actually makes them connect in some way. And it's not just behavioral connection, it might be actually neural connection. So again, there's a lot to be discovered, but there are lots of surprises here because we're starting to look at brains in a naturalistic context. And here's one last surprise, and this is close to my heart. This is work by Camille Testard, who was a graduate student at University of Pennsylvania. And this paper, on which she's first author on it, is about to come out in Nature in the next couple of months. And I was the chair of her thesis committee. And what she basically did with her team, and again, all this type of work is impossible with one individual. You need to work with teams of engineers, is you have a number of different macaques, in this case, interacting in these cages. So in the left panel, you see a green macaque and a purple macaque and an orange macaque, and you can present them in different situations, either alone or paired with a male paired with a female, or male paired with another male. And then you can record from different areas in the brain. And so you can see a brain from the one monkey that's interacting, that's monkey A, with another monkey H, that's interacting with, and we can have, in her case, they use wireless probes that have like 100 electrodes you can pop into the brain, and you can record from many, many neurons simultaneously. And it's very hard to see probably the size of the screen, but you can see the dotted line here, and the bottom panel, it's sort of all blue with some yellow. Yellow means the neurons are very active. Blue means that the neurons are not very active. And what you can do in the top panel with all the different colors, kind of the rainbow colors, this is, you can quantify the types of behavior that these animals are producing. Are they grooming with each other? Are they fighting? Are they showing aggressive interaction? Are they vocalizing? And so on and so forth. This is called an ethogram. And you can look to see when is activity active in these different brain areas. And in the red dotted line, you can see that both the top area that they recorded from, known as TEO, which is part of the ventral stream in the visual system in the temporal lobe, as well as the prefrontal cortex on the bottom, they're very active during this red little bar right here in both cases. And the red bar is aggressive interaction. And this was a surprise to Camille because she chose two areas. One in the prefrontal cortex that she thought might be influenced by the social interactions that the animal's producing. And then she chose a control area. And the control area was this area TEO, this part of the visual system, assuming that this area would only be active during visual interactions between the two monkeys. And what she sees here is that both areas are active during specific types of social interactions, really going against the idea that areas in the brain are only specialized for one specific task and not another task. In other words, social interactions almost seems to override what these areas do. And so for me, this is incredibly interesting, because if you look at all the early work that was done in anesthetized preparations, is that we are sticking electrodes and we're saying, okay, is this area going to respond to a visual stimulus or an auditory stimulus? And in the early days, we had areas that were only responding to either visual or only to auditory, but it was a passive presentation of stimuli. Now we're having the animals kind of interact with, in a complex environment, and you're recording from a visual area, but it turns out that visual area is maybe not just visual. And the analogy I want to give you, because this has happened to me several times throughout my career, which I think is interesting, and I think it happens throughout biology. I'm calling it the FGF analogy, FGF standing for fibroblast growth factor. So when I was in grad school, I looked at the effect of growth factors, or also known as trophic factors, on the growth of neurons in a dish. And I found that one of the best growth factors to make my neurons grow, these were ciliary ganglion neurons from chick embryo, was the fibroblast growth factor. And fibroblast growth factor was discovered as a growth factor that made fibroblasts grow. But it turns out that fibroblast growth factor is all over the brain, and it actually controls growth and retraction of many different areas in the nervous system. And so the name really throws you off, because you just assume that fibroblast growth factor is only for fibroblasts. In fact, it's mostly for neurons. So had it been first discovered in neurons, it might have been called nerve growth factor. So I think we have to be very careful with semantics and labels, and not let ourselves be biased too much by prior labels as to what things are supposed to do, because they might not always be doing what they're supposed to do. So again, many advantages of looking at animals in a naturalistic social context. There's some surprises that are going to emerge. That's certain. We're going to have to maybe redefine the function of certain brain areas. But there are also challenges, of course. One, and I put it in bold and underlined it, I should have put it in a bigger font, is behavior is highly variable. So that's going to be very, very hard, right? The birds are singing and then they're responding, and the context is different every time. The song might even be slightly different every time. The responses might be different. So it's highly variable. How do you make sense of that? Another and second point is one that we're trying to grapple with, is social context. So what I mean by social context, for example, if I'm talking to an individual, that the context is just that individual talking to me or listening to me. But if there are other individuals around that person I'm talking to, that is now a different social context. Some of these individuals might be my friends. Some of these individuals I might have just had a conflict with several hours before. We might be competing for the same female. We might not be competing for the same female. Who knows? That's the context. And I have a sense that context is really important in determining how the brain responds to different social signals. And so the question is, how do you quantify social context? And I think that's a really important variable in neuroscience that no one has really grappled with. And we're trying to do it. When I say we, it's really, the computational people that are part of the team. Other challenges, of course, are going to be technical, and these are going to be ongoing. How do you record neural activity in a wireless fashion over an entire breeding season? How do you do behavioral tracking? How do you analyze all that data? And then, obviously, these are techniques that are very high level, very sophisticated. How do you build your team? And how do you integrate? And in the Q&A, I'd be interested in looking into these types of questions. How do you integrate across disciplines? It's not easy as a biologist necessarily to talk to a computer vision expert. They know things they assume I know, and I know things I assume they know. And so how do we create this dialogue so we can have a really good interaction and be productive? So what I want to do today is, this is my outline. I want to tell you a little bit about why I personally got interested in recording in a naturalistic condition. And then you might ask yourself, yeah, there are lots of animals that are social. Why are you choosing songbirds, and why specifically cowbirds to do this? And then I'll tell you a little bit about the smart aviary we built, kind of showing a little video about what it actually looks like, and then describe a pipeline of how we acquire the data, or plan to acquire the data, and some of the questions we're interested in. And then I want to just finish off by giving you 3 challenges that we have faced. One is kind of a funny one. It's actually not funny at all, but it is sort of funny and with hindsight. And then 2 that are more, can be generalized for this type of approach, which I think are really important. And then I'll briefly conclude after that. So this is my personal journey. So I was recording in birds. I was one of the first in my field to record in awake behaving birds. I showed you the picture where the birds had the little wire sticking out, and they were free to move, and they were singing happily, and we could present them to a male or to a female. You could present auditory stimuli to them through a speaker, and then you could do that when they were awake, or you could turn the lights off and then have them fall asleep, or we could even anesthetize them, and then ask how did those responses change. And on the right top panel, I have a brain. This is part of this neural circuit for birds. I have one electrode in this area called HVC. It's a motor area. The motor circuit is shown in green, but it also receives auditory inputs shown in gray. And so we can think of this as an auditory motor area. And I stuck here an electrode in HVC, and then also in, we call it auditory forebrain, but if you want, you can think of this as auditory cortex, so a higher order processing area. And when I present a song, this is just passively presenting the song when the animal is resting, and resting could be it's anesthetized, or it's sleeping, or it's just groggy. What you notice is that the HVC shows a very, very strong response. So you can see before I present the song, there's not much activity in HVC, and then there's huge amounts of activity, and then it goes away when the song is no longer presented. And you get the same level of activity in the auditory forebrain. So nothing really interesting. Basically, both areas of the brain respond when you present song. But what I was really surprised with is what I'll show you in the next slide, is that when now I woke up the bird from this groggy state, or maybe the lights were turned off, he was maybe sleeping, and now the lights are on and he's alert and looking around, when I presented song, I get a very strong response in the auditory forebrain, similar to what I saw before. But, and this was so, almost fell off my seat for this one, when you looked at HVC, is the response were completely suppressed when you present a song, when the animal transitions from being asleep or just resting to being awake and aroused. And so we spent a lot of years, shown in the papers on the bottom right, trying to figure out what the mechanism was. And we found that norepinephrine, which is a neuromodulator in the brainstem, has a very strong projection to one of the areas that interfaces between the auditory forebrain and HVC. And it's actually called the interface nucleus. And we found that norepinephrine is responsible for this. So if I were to remove norepinephrine in the aroused state in HVC, I would now get a response. So what that told me is something I think very important, is that there are areas that are pretty stable in representing the world, but there are other areas such as HVC that are representing the world differently depending on the animal's behavioral state. Here the animal is showing an on-off response, and we call it gating. But anytime you invoke a neuromodulator, that means that it's not a gate, it's really like a gain control. You can turn on the response or turn it off, or just have it just on a little bit, depending on the context. And this is when I thought, wow, it would be so cool to be able to look at how might these auditory responses change as a function of social context. So why songbirds? So songbirds communicate mostly with vocal signals. So males in almost all species of songbirds sing a song. Females in some species sing a song, but mostly don't. But they still vocalize. They'll produce calls. Zebra finches on the top right panel sing a very precise song shown by these 4 panels to its right. These are known as sonograms, and their song is shaped by a sequence of syllables, A through G, and like and they sing it over and over and over again, and it's very, very stereotyped the way they sing it. Using the zebra finch that sings a single song, we've basically, my field has delineated an entire neural circuit shown on the left in the brain diagram that I showed you a little bit before, that is responsible not only for the production of song, but the perception of song and also the learning of song in a reward manner that's based on internal rewards, and there's dopamine that's involved. And all these brain areas have specific names that are specific to the songbird, but they have their homologs or analogs in mammals. And so one of the advantages of songbirds, obviously, is that they're vocal communicators. If you have a vocal signal, you can quantify that really, really well. And because this is important for social interactions, we know exactly when a song started, when a song ended, and we can quantify precisely the structure of that song. If you look at other animals that are social communicators, let's say monkeys, a lot of their social communication is through preening. And so they're touching each other and they're applying different levels of tactile force on that individual. And that's really hard to quantify. You don't know when they started pushing a little bit harder, certainly externally, and how that might affect the individual. So I think birds are really powerful because the main signal to communicate socially is through acoustic means. On the bottom right are the cowbirds. One of the reasons I chose them is because they're much bigger than the zebra finch. So I knew that I would have to develop technologies to put on their heads, and they weigh about 45 grams, whereas the zebra finch only weighs 13 to 15 grams. So there are advantages. It turns out that the male cowbirds sing, the females don't. And it turns out, we were excited about that the male cowbird song is just as stereotyped as the zebra finch song, which offers us a lot of advantages for the types of questions we want to try to ask. So in addition to using social signals, here are the social signals. Oops. These animals that we chose cowbirds, I told you in part for the size, but cowbirds are incredibly socially gregarious. And a lot of birds are socially gregarious, but I think one of the reasons the cowbird is very interesting, it's actually a brood parasite. So the female basically dumps her egg in a nest of a host species, maybe a warbler, and when the eggs hatch, the new hatchling has to basically ignore everything that mom and dad tell it, because that's not his mom and dad or her mom and dad. But once they leave the nest, once they fledge, they get together in these huge flocks, and a lot of the learning happens there. A lot of the social rules happen there. So they live in these large flocks, and then they kind of spread out and segregate when they have to find nests to parasitize. Paradoxically, the cowbird also pair bonds. So there's no parental investment. As I mentioned, they're brood parasites, but they do pair bond. And pair bond is essentially a marriage. And this can last for many different seasons. So the male is married to the female and they stick together. And so there's a whole buildup to what actually makes a female choose a certain male to actually be her mate. And this is a really important point in the timeline of courtship and reproduction that we are very interested in. Again, we're interested in studying behavior and neural correlates of behavior within an evolutionary and behavioral context that is ethological, that is behaviorally relevant. In addition to them being social, they also exhibit really interesting behaviors in the aviary that we've shown. This is a paper that we're working on right now. Actually, before I go to that, let me tell you one more thing, because I jumped ahead. So I told you that the cowbirds have social signals. What they also do is they have also really interesting postural signals that are part of their repertoire of social communication. So in the left of my panel, you see one male bird singing to another male. These are brown-headed cowbirds, so the feathers are black and the feathers on the head are brown. And you can see there's this display that the male produces when he's singing to the other male. In the middle, when females really like the song and really like the male, they'll produce this arching of the back. It's a lordosis-like response. It's called a copulation solicitation display. And this allows the male to eventually mount her. And then there are displays shown to the right that are so-called head-up displays. So when a male is very aggressive towards another one, they'll walk up and kind of lift their head and walk towards them. But there are other behaviors that we only see in these complex social environments. This is a very interesting one. No one knows what it is. These birds do these head down displays where they'll come together and basically hold their heads together and be completely immobile, sometimes for up to 2 to 3 minutes. And we don't know if this is an aggressive display or a submissive display. We just don't know. And then lastly, one of the behaviors I'm really interested in is this very subtle behavior. Here's to the right, on the bottom, there's a male singing, and the top left, there's a female. And you can see her wing is lifted a little bit from her body. This happens very quickly, within about 150 milliseconds, and this is known as a wing stroke. And we think this is really important in females, telling the males whether or not they like them. I realize I'm running out of time, so I just want to tell you real quickly that we see in these groups interesting group-like behaviors that can actually correlate with reproductive success. So if males to do their behaviors in a cohesive manner, that aviary tends to produce less eggs, more eggs, they're more successful. But if males are not cohesive in their behaviors and kind of all over the place, then that aviary tends to produce less eggs. So building our smart aviary. So we started that with a large grant from NSF. So we built this aviary. I mentioned it to you before. It has 10 computer vision cameras and multiple microphones. And this is roughly what it would look like if you were to image that. So here in this video, landing on one of the perches and singing with the display, and I think there was just a male on the triangular perch on the right singing. So you can see that birds are moving around all the time, and so our challenge is to try to locate the birds, locate who is actually singing what song, and be able to make some sense out of that. So that's the challenge, and it's a pretty large challenge. So what we've decided, I'm showing you here, is a pipeline. So we have from our 8 to 10 cameras, they were sampling 40 times per second. We use a lot of artificial intelligence tools, so neural networks, to actually take these raw images and turn them, as you can see in the second panel, into different blobs. And each blob has to have a different color because it's going to be related to the identity of each individual. And then what we need to do is, birds, as you saw in the video, are not just stationary, but they're moving around. So we need to be able to track them throughout time as they leave the perch and actually fly. And this turns out to be an incredibly complicated ask from engineers. Most of the work that's looking at tracking in animals is done in 2 dimensions. So it's maybe you have a camera on the top looking at 2 mice interacting in a box. But here you're operating in a 3-dimensional, in 3 dimensions, with birds moving really fast. From start, takeoff is very fast. And so you often miss many frames if you're sampling at 40 times per second. So once you can track them, then what we want to do is we've developed this pretty cool method known as an articulated mesh method, where we can take a bird shown on the bottom left, and we can detect key points automatically through neural networks, and we can fit this articulated mesh, which is shown in the blue panel, on top of the bird, so that matches pretty nicely. And you can see, and I think in the next slide, no, it's not shown. There were a few birds that could move around. Basically, with this articulated mesh, we can actually, let me actually go back to that slide. We can do is we can capture the shape of the bird. And the idea is to be able, using this ability to capture pose of the bird, is use classifiers to eventually classify whether a bird is producing a display when it's singing, or when it's producing a head-up display, or whether a female is producing a copulatory response, or eventually a wing stroke. And so we can classify this and then turn these videos that have tons and tons of information into select points where we can know the individual's ID, know its position, and what it did at a specific time, so we can track moment to moment what every bird in the group is actually doing. Obviously, sound is very important, so we have an array of microphones, and here's an example on the top right of 2 males singing at the same song. One is singing the orange song, one is singing the blue song, and through our array of microphones, you can see on the bottom right panel, we can create probabilistic cloud of where the sound actually came from, and we can identify, if you look at the cage on the left, that the bird what's surrounded by the orange is the first male that sang, and that's where the song came from. And then the bird in blue sang the other song that came later, and he was in a different part of the cage. So we can use these sophisticated methods to pinpoint the location of sound, the identity of the birds, where they are, and how that changes over time. The goal, and this is a lofty goal, is to take all this information and create a giant array for every single bird, for every single moment in time, and ask, where is it in space? Maybe who is it looking towards? You can look at the gaze based on where the beak is, and what kind of behavior is it doing? And then you can build these types of representations that I'm showing you to the right. It's known as an ethogram, where you have, in this case, obviously we have 16 birds, but in this case, I'll just show you one female and two males. And you can see the female might have rattled once, and then she's preening, and then she rattles again, and then she copulates. But why did she copulate? Well, it turns out that male A, who was showing an aggressive display, all of a sudden flew towards her and then sang a bunch of songs. And then the third song eventually elicited the copulation. So we might want to, as neurobiologists, ask, what was special about that third song in that string that elicited the copulation? So we might want to record from the female as she heard that song, and how did her brain respond, and did it respond differently to that song than prior songs that actually led to that copulation? And is this possibly due or modulated by the social context in which she finds herself? So how do you record? Well, Firuz Aflatuni is a very incredible electrical engineer with a great degree of confidence, and we're building these chips that are about, the chip is very small, 1 by 1 millimeter, and it sits on a PCB board that's about 5 by 7 millimeters. And you can see here, it's directly attached to electrodes. And the idea is to implant these in the brain of the bird. And this is so small that we can cover the skin and the feathers back over it. So every bird in the aviary will look like every other bird, except that we can record from these birds throughout the entire season, because these chips are wirelessly powered, and they're also transmitting in a wireless fashion. And we can ask these types of questions. For example, on the panel on the right, we have male 1, M1, singing to female, F1, and in 3 different contexts. In the top context, it's just male 1 singing to female 1. In the second context, male 2 is present. And in the third context, it's different females present. So that's the immediate context within which you record the response. So did the neuron respond to the song of male 1? But what you want to know is, you want to build something about the social context. You want to know what happened a few minutes before. Did the female just receive a song from a different male? Or did she not receive a song from a male? Or did she receive another song from male 1? But then we want to know who is male 1, who is male 2. So we want to know a little bit about the hierarchies. And so if you go back in time and collect over weeks, you can tell that male 1 is dominant over male 2, and that male 1 is pair bonded to female 1. So these are the types of questions we want to be able to ask. So challenges. And so these are my last 3 slides, and I'm going a little bit over time, but these are, I think, interesting, and I would like to be able to finish with this. As I said, some are really annoying challenges. This is the most recent annoying challenge, but it's sort of funny in hindsight. This is our aviary in the left panel, and it was right beside some piece of land. I work in the city, I'm in Philadelphia. That piece of land was very quiet for many years when we built the aviary. It turns out that is a site where Penn dumps all extra dirt from their construction projects, and they have to sift and remove big pieces of piping and concrete and dump it in these big metal containers, which creates a huge amount of noise. And so we solve this now by providing this very expensive 12-foot high fence with acoustic attenuating curtains. And fortunately, university was able to pay for this. This was what I spent most of my fall trying to figure out how to do. But there are other more interesting challenges. This is one that is, I'll call it dream versus reality. What the biologist wants and what the engineer can deliver. And I've had many conversations. So I'm the biologist, and I say, I want to be able to collect behavior from every single bird for 6 hours per day. And I want to do it from March 15th to June 1st. And then the engineer comes to you and says, Mark, why do you need all this? You're getting about 15 terabytes of data per day that's compressed. And this is very, very difficult to manage. Where are we going to put it, and how are you going to analyze all this data? So these are the important conversations that the biologists need to have with engineers. There's the dream, that's fine, but then there's also a reality check. And so in the end, what we've decided is to actually only record for 2 hours a day and maybe only sample every few days. Things don't change so fast. And might we miss something? Yes. We also decided, do we want to track birds in 3D all the time? No, we probably just care about what happens on the perch. And so that helps us a lot. The last challenge I'll talk about is finding a solution that will work. And here it's interesting, it's the opposite. It's what the engineer wants versus what the biologist needs. So the engineer, if you get a grad student in computer vision, they want to do cutting edge engineering that will get the PhD student to produce something that's worth publishing. As a biologist, I just want something to work. So as a quick example here, these birds fly in 3D. It's very hard to track them in 3 dimensions, as you can show in the right panel. As 2 birds identified, blue in A and red in B, as they cross, now you lose the identity. So you no longer know. It's very hard to know who is A and who is B. And these automated systems make big mistakes. So the engineers spent a whole year, and this was my mistake. I totally trusted them. It's like, oh, you're engineers, you guys are magicians, you can do anything. They spent about a year trying to identify the bird based on the color of their bands. And in this array chart here, you can see it's just all these numbers, but it's like, oh, we can tell you that with 25% probability, it's probably Bob, or with a 38% probability, it's actually John. And this is not very useful for us. So we've actually modified, and we now have all our birds have bands with little radio frequency tags, and we're trying to get identification that way. So quickly conclude, neuroscience in a naturalistic environment offers exciting possibilities, but it also offers many challenges, both technical and conceptual. I think it's really important in this type of approach to actually choose well-defined questions, because otherwise you'll be overwhelmed with complexity. And what we've chosen is to really focus on how the brain responds to song signals, acoustic social signals, and how they change with social context. And then the third important point is that this type of work requires transdisciplinary teams. And so that means building a bridge across fields and really understanding the limitations and goals on both sides to be able to come up with something that's actually fruitful and productive. So I just want to thank my funding NSF and all my colleagues. And I want to go back to you, Christine.
Mark, thank you so much for that excellent presentation. It is now time to start our Q&A portion of our webinar. Now, to our audience, if you haven't submitted any questions yet, please do so now. Just type them in the Ask a Question box, click Submit, and we will answer as many of your questions as we have time for. So, Mark, we already have some great questions coming in, so if you don't mind, I'm just going to dive in.
Yes, please.
Yes, let's start with this one. It seems like position within the social hierarchy would be an important variable when determining social context. How do you measure hierarchy in males and in females?
That's a great question. So let me answer the one for males, because that's easier. But it's not trivial. So the way we measure hierarchy in males is we look at song interactions, because that's something that we can measure. And this is something that other people have done. So for example, if I'm singing to another male, if I initiate the song, so these males do these directed song displays. I'll sing a song, and then the other male will answer, then I'll sing, and then he'll answer. And we go back and forth in these long bouts. And we can look at who is singing to whom. And based on those bouts, we can establish male hierarchies. So that's reasonably straightforward. I think in neuroscience and in science in general, certainly in this field, people are focused on males because it's easier. Males sing. But females are probably way, way more important than males in these. They're making all the important choices, but they don't have easy behavioral measurements to actually track hierarchy. You might maybe look at aggressive interactions between certain females and maybe establish hierarchy that way. but it's really difficult, and we don't know. And we're hoping with this data set that we can maybe extract that information about female social hierarchies based on, who's choosing the best males, and if we can, say, okay, the best male is chosen by that female. What did she do to actually to attract that male? And maybe that means that she's sort of the highest on the hierarchy. And then we can look at the behaviors. Maybe she was the most aggressive, or maybe she was the most, I don't know, maybe most exploratory. It's hard to know.
Thank you, Mark. Our next question: in bird courtship, is the coordination choreography between the male's song and body movements important in the female's selection? For example, do females prefer males that can sing and dance at the same time?
Yes, that's a really excellent question. And we don't know the answer to that, but it turns out when the males sing, I told you they have this wing display, right? They kind of produce this big wing display. And when they sing to other males, they actually pump their wings at very precise times in the song. And those are synchronized to when little gaps, when birds take a little breath between syllables in the song. But it's interesting because when the male sings to the female, they actually almost, it seems like they're trying to suppress this wing spread. So when they sing to the female to try to impress her, they're not doing that. And we don't know. It seems paradoxical. You'd think that the female would want the male to kind of show all its wings and all its display, and that's what she's evaluating. We think that it's a little bit like it's a variant of a hypothesis that's been presented in behavioral science and behavioral biology. Is called a handicap hypothesis. So let's say you have two skiers. Let's say Christy and I, someone asks us, do you ski? And we both say yes. If someone asks me, are you a good skier? Say, oh yeah, I'm a great skier. Someone asks Christy, are you a great skier? Oh, she's a great skier. But how do we know? So we'll go on a bunny run and we both look great. Okay, fine. So think of that as bunny run. You're using the poles, you're using your wing spread. But now we put us on a bump run, this little bit icy, and we take the poles away and we tie our hands behind the back. That's when you can truly see that Christy's much better than I am, for example, right? Because she does not have to cheat using her poles. And we think that maybe the males are the best males are those that can produce really high quality songs without using their wings.
Great analogy, Mark. Next question. Do you try to mark the birds in different color to make it easier for the computer vision?
So this has been proposed many, many times. One of the things that's very tricky with this type of approach is that you don't want to mess up with the normal ethology of the birds. So we know, for example, in zebra finches, that the color of the beak is correlated with testosterone levels. So males with high testosterone have much brighter beaks than males with low testosterone. In starlings, you notice during the spring they have yellow beaks, and in the fall they have black beaks. So it's yellow because of high testosterone. So color has meaning. So if we start messing with color, it might actually change relationships between birds. So we have to be very careful. But we do have bands with colors. We actually moved away from that. So now we're putting little radio frequency tag in the bands. We 3D printed these bands. And so the radio frequency tag can actually give us identity. So we don't have to add any color. And that's the other reason we don't want to put anything on their head, because imagine if you have something sticking out of your head, it might actually change how you're interacting with other birds. So we're trying to keep everything as natural as possible when we do our behavioral and neural recordings.
That's fascinating. Comment: wonderful talk. Might there be discoveries that might be generalized beyond birds? And what could be a framework for doing this?
This is a great question. This is something that, my parents' friends ask me all the time. It's like, how can we make money out of what you're doing? Or how is this relevant to humans, right? And it's a great question, and it's always a challenge. So I think what we're looking for is basic principles. And these brain circuits have evolved to encode social information. Are the brain circuits in birds homologous to those in mammals? No, but they derive from the same tissue, and they have, many of them are probably analogous, right? Certain analogous, even homologous, possibly. So if you look at just basic brain function and how the brain processes social information, I think we can extract from that. And I think that that's the hope. And it's a hard question to ask, but I think one of our advantages with birds is the social communication side. So we have these acoustic signals, and we can look based on the precise onset and offset of the song. We can look at when the brain actually responds, whereas many mammalian species that are studied either are not social, like all of 90% of neuroscience studies mice, they're not social. They might have dyadic interactions, but they don't have higher social interactions or rules. And animals that do have complex social rules and dynamics, like many monkey species, it's going to be hard to study social behavior because you don't know when they're starting to groom their mate. That's one of the major social signals.
Interesting. So, Mark, can you speak to how understanding these complex behaviors in birds can help shed light on human behavior, such as understanding hidden states, emotional states, etc.?
So that's related to this other question. I think it's a great question, and I think certainly in my sort of next 5-10 years, I probably won't get to that. I think identifying basic properties of how the brain might encode social information is the best we can do. To actually encode sort of internal states, we might need more of an ability to manipulate the circuit with a lot of the tools that are being developed in rodent models. You can use viruses to activate or turn off certain areas. Certainly that is something that's going to be interesting, is being able to manipulate circuit activity and ask how does that impact behavior. So for example, I'm not answering the question directly, but I think it's an interesting thing to think about. I can record neural activity in a social context and ask how does social context impinge or impact how the brain responds. But what I can also do is I can shut down different areas of the brain, which I can sort of online modify the behavior of a bird within the group and ask how does that impact behaviors of others or the entire social group towards it. And so we had a really interesting finding a while back where we lesioned this in a small area in the female, and the female seemed completely normal. But then we noticed that the males, who normally only sing to their wives, basically, all of a sudden all started singing to these females that had these small lesions. Somehow she was emitting some signal that was very subtle that males were catching on. And so just this small lesion basically destroyed the social integrity of the group, with having males all starting to sing towards these new females. So the manipulation of brain circuits, I think, is going to be a very important tool. And the more we can understand the circuitry and manipulate specific nodes in it, we can get to things like hidden states. But that's a great question.
Now, Mark, we're almost out of time. So we're going to wrap with this question. It's several parts. So I'm going to read the whole question, then if you can answer part by part.
Great.
To what extent are the approaches, algorithms, and computational analytics used in your aviary similar to or built upon 2D systems used in Drosophila and rodents? To what extent are the tools you developed portable to other species contexts? And does each investigator have to develop de novo tools, etc.?
Yeah, so we definitely learn a lot from work in rodents and fruit fly Drosophila, right? So there are lots of packages out there like Deep Lab Cut, a package called Sleep that we're working with, Sleep, SLEAP, that was developed at Princeton for tracking animal behavior. And so we are developing our own. So this was the mesh model. It turns out that mice, and you can sort of skeletonize a mouse. Birds are tough because of the feathers, so they puff up their feathers, and so there's a whole shape. I also think of birds, the wings in birds, it's kind of like their hands, right? I'm talking with my hands all the time. I think that wings are very hard to model because there's so many different articulations. So these sleep systems don't really work very well for that. So we have to sort of develop our own. We're actually challenged right now to try to figure out how to do that. How do you model a wing? So in terms of the analytics, I think that we don't have a big data set yet to really work with, but that's going to be kind of where my colleague VJ comes in out of physics. And we're using these models borrowed from statistical physics, like Ising models, to look at every possible interaction between every individual and look for some sort of higher order social organization. These types of methods that we can apply to this and innovate are absolutely going to be applicable to other social structures for sure. And that's certainly the plan. And I forgot the third part, but yes.
Does each investigator have to develop de novo tools, etc.?
So to some extent, yes. So I think one of the things that I realized is that a lot of the tools exist off the shelf, but you can't just buy them and put them together, right? So the computer vision cameras exist, but integrating all the computer vision cameras with the microphones and the RFID tags into one single pipeline is very complicated. How do you compress all this data? So, for example, the operating system we use is called robotics operating system, which is very specific, right, and niche. And not many people know how to code in that, but that's really important because that's the most efficient way of actually collating or integrating lots of visual data with microphone data. And so these are yeah. So to some extent, there's a lot of development that has to happen.
Mark, thank you for this fascinating talk and informative Q&A. Do you have any final comments for our audience?
I was just loving the opportunity to speak and to kind of get you excited about model systems that are different from the mainstream. And I think we can learn a lot from studying birds and from studying bats and frogs and all sorts of animals.
Well, Mark, just based off our Q&A and the questions that we weren't able to get to, you definitely have struck everyone's interest and excitement. I do want to thank you again for your time today and for your important research. And before we go, I do want to thank our audience for joining us today and for their interesting questions. Now, those questions that we weren't able to get to today, and additionally, any of those that come in during the on-demand period, they will be answered via the contact information you provided at your time of registration. Today's webcast can be viewed on demand. That's for 1 year through March 6, 2025. And Laboratories will alert you via email when today's webcast is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. Mark, thank you again for joining us. And thank you, everyone. Have a great day.
Thank you. Appreciate it.
Bye-bye.